Okay, thank you very much for the introduction, and thank you for the funding that Jesse's Journey has provided for our research endeavours in the field of DMD. I want to tell you a little bit about my career and a little bit about what we do, and that goes back to the very basics. So I gave this talk for the mayor in Mississauga, and it was an accidental career in cancer research, and now it's an accidental career in DMD as well. And I can give you a bit of background to this, which really this is the most bizarre entry point into a field of research for me, but it's been a really fruitful one and one that we're really excited about. So over the last 10 years we've been developing small molecule inhibitors of proteins that are involved in driving human cancers, and really fortuitously it turns out that those same proteins are actually activated in DMD and play a transformative role in DMD. And this wasn't really realized until really about 6 months ago that we've figured this out, and I'll tell you a little bit about it. So a brief bio: I'm not a physician, I'm a chemist, I'm an organic chemist. I studied chemistry at Glasgow University. I didn't want to be a chemist either, I wanted to be an architect. I love painting and drawing. I didn't really like chemistry that much, but I ended up going into it because I didn't want to be at university for 7 years studying architecture, and chemistry was a better option. It was only 4 years. So funnily enough, I ended up deciding to go to the University of Glasgow, and I wanted to go to Glasgow because I loved architecture and I loved art, and I loved the Barry Gothic building of Glasgow University, this wonderful building which looks a bit like Hogwarts, which is ironic because all my students, every single year, we get these forms to fill out. The students critique you, but they don't really critique me, they just tell me that I look like Harry Potter. So I just get these messages every single year since 2008 saying Gunning looks like Daniel Radcliffe. I don't. Gunning told class he doesn't look like Harry Potter, but he should know he does a lot. So this is what I look back like way back then. So there's no real similarities. Maybe a little bit, but not anymore. I grew a beard, so that worked out really well. So you really look like Harry P in 2016. So at Glasgow, I made molecules really for the sake of making molecules. I wanted to make complex molecules that nobody else had made for the purposes of making something that nobody else had made. There wasn't really a goal behind it other than to make something that had never been made before at an atomic level. And it wasn't until I went to the US where I was charged with making inhibitors of specific proteins. That is, to make molecules that bind to a protein and inhibit its function in human disease. And I was charged at the time with targeting this protein. This is signal transducer and activator of transcription 3 protein, which is a mouthful. So we call it STAT3 for short, and it will shorten this presentation by saying that too. So as was mentioned, I come from a little island on the west coast of Scotland. So I'm actually from Paisley, but I lived my entire formative years on the Isle of Bute. So this works for this story. And it's identical to STAT3. So maybe it was fate that I was meant to target this protein. So I'm going to tell you a story about making drugs, legal ones, not like crystal meth from Breaking Bad. My students would never do that, ever, and they would certainly not profit from it. This was for a TED talk, and I said to my students, you need to do something Breaking Bad. So they took copper sulfate and froze it, and then they had this little bag. So STAT3 has been coined a master regulator of the cancer cell. It's been shown that in cancer it's hyperactivated, so it's permanently switched on. And in fact, in 70% of all cancers, this protein is turned on. So It's been shown that knocking out this protein actually leads to a cell death event. So from an oncology perspective, inhibiting this protein was seen as being a viable cancer therapeutic strategy. So to give you a very basic sense of what I do, why do drugs work? So to understand this, you have to think at the highest levels of educated society. You have to think like a chemist. And chemists view the body not as a bag of muscles and tissues and organs. We view it as a large reaction chamber, which is full of molecules, large and small. And these molecules undergo complexation events and chemical reactions to facilitate the biological processes that allow a human to function. For example, serotonin, this very small neurotransmitter, bridges the gaps between the receptors, your neurons, binds to receptors in the brain and in the stomach to control your emotion, how you feel, whether you're positive or negative. The key fact is that drugs interact with these large molecules to modulate the way in which the body works. So what is medicinal chemistry? At its most fundamental level, it's the design and synthesis of small molecules which bind to a target receptor. And the consequence of that interaction is a biological response. So if you choose this receptor properly and you make a molecule that binds to it properly, you will lead to a biological response. In cancer, that response is to kill the cells. Why do you need to be selective? You need to be selective because your target is not the only target inside the cell. There is millions of other targets that you can potentially interact with. Why do you need to be potent? You need to be potent because if you're administering a kilogram of drug, you're going to have off-target effects, whether you mean to or not. So, to give you a sense of selectivity, an average drug dose is 10 to the 18 molecules. That's a lot of molecules. The average human has 10 to the 15 cells. Each cell has 10 to the 10 molecules. So when you look at it, you have a 1 in a million chance of interacting with your protein. So when you do bind to that protein, you have to stay there long enough to engage and elicit the biological response. So where is our project? What is it based on? Our molecules actually target not just a protein, one individual protein, but it targets a protein complex, the STAT3 complex. And this is the biologically active form of this protein. And our goal is very simple. Can we make molecules that come in and displace, break open this protein-protein complex? So this protein interaction binds to DNA and promotes transcription of genes that drive both the cancer phenotype and, as it happens, DMD. So where do we take inspiration from to try and design molecules to do this? So this is the crystal structure of STAT3, and you can see the DNA bound in the middle, and you can see this yellow portion of the protein. This yellow portion facilitates that complexation event. So the idea was, can we use that particular part of the protein to inspire the design of small drug-like molecules that interact better with the protein and outcompete the other protein, so effectively disrupting it. So it's a bit like a jigsaw puzzle. So if you look at that area of the protein, you can see that there's 3 main binding sites within the protein active site. And the idea is to try and make molecules that can fit within those 3 sites and bind to it with beautiful complementarity and potency. So the first molecule that we discovered, can you see my, yeah you can. This molecule here, we'll call it compound 1, has an IC50 of 90 micromolar. So the first question you're going to ask is, what's an IC50? An IC50 is the concentration of drug required to kill, in this case, cancer cells, 50%. And it's a bar to give us a sense and to rank and stratify our molecules in terms of which ones should go forward. So 90 micromolar sounds like a very small quantity of drug, and it is. But in actual fact, to have a clinical efficacy, you need to have an IC50, which is closer to 0.5. So you're looking at 180, 200 times less drug to kill the same number of cells. So when you look at this, the way in which this molecule binds, you can see that this molecule was derived from computational screening, and it binds to 2 of the main sites, but this upper portion is completely free. So the idea was that we could adopt this structure and optimize it so that it could better fit this active site. And to do this, we needed a piece that would fit within this upper portion of the protein. And to do this, we decided to build off of the nitrogen in this molecule here. So, simple goal is make molecules that fit this protein better. And by doing this, we're tailoring the molecule to the protein. It should therefore have higher affinity, you should require less of it, and it should be less toxic and more effective. So to do this, we make molecules. So despite all the supercomputing power in the world, and we actually have now IBM Watson in our lab. So we're using Watson, they're trialing it in our lab to see whether it's of benefit to chemists. So we have artificial intelligence driving this as well. Despite that, it never works first time, ever. So what we do to compensate for that is we make lots to de-risk the project. So we do this using organic chemistry, which is everyone's favorite subject at university. Biologists call us cooks. They think we just mix things together, put it in the oven or a round bottom beaker with a Bunsen burner, and out pops something else. It's because they have an inferiority complex that we can manipulate matter at the atomic level and we can turn back time. So the next step is to actually synthesize these molecules. So to do this, it takes, in this case, with the family of compounds that's going to be used in Duchenne, this is an 8-step process to make one molecule. In the history of this project, about 900 molecules have been made to get to the ones that are the most active. And each step in this process takes about a day. So it gives you a sense of the amount of hours that PhD and postdoc trainees are spending to make just one of these. So we make our family of compounds. We'll denote it with this little bunch of shapes. So what's the first step in a medicinal chemistry program? We first have to test whether the drug binds to the protein outside the cell. If it doesn't bind outside the cell, there's not a chance in hell that it's going to bind inside the cell where there's this other million potential targets. So we screen our family of molecules against the protein target, and what we're looking for is 1, 2, or 3 molecules that bind with high affinity. Once we have that, what do? It comes back to selectivity. We screen against similar proteins and completely dissimilar proteins. And the goal there is to hopefully see that our molecule is fidelitous to our target protein, that it's not promiscuous and it's not binding to lots of other proteins. Then what do? We put it into cells. So this can be Duchenne, muscle cells, or we can look at, in this case, cancer cells. So this is breast cancer cells that have been treated with our drug. And what we're looking for is efficacy. We're looking for a phenotype that is positive. So in cancer cells, that's death. In the next step, we then look at healthy cells. Again, selectivity. It doesn't matter whether you kill cancer cells. It matters whether you don't kill healthy cells at the same concentrations. So I can kill cancer cells with bleach. It doesn't mean that that's going to be the next therapeutic drug. And the goal is that we want to see a therapeutic window between the 2 different disease and normal type cells. We then go into the cells, we do molecular biology, and we want to see whether the target has been engaged. What does that typically look like? It looks like a decrease in the amount of target with increasing dose of our drug. And then what do? Selectivity again. I sound like a broken record, but this is the process by which medicinal chemists work. And what we want to see is that you don't have an effect on other proteins, that your effect is limited to the target. And then we go into animal models. We either go into models that have been engrafted with human tumors or DMD models, mouse models. And what we want to see is 2 things. 1, efficacy, that we hit our target and we have an effect on the disease? And 2, does the mouse have a stroke as a result of taking our drug? Do we see necrosis of the liver? Do we see heart problems? All these things. Do we see weight loss, another indicator of severe toxicity? So what have we done at U of T? So we started off with this compound, compound 1 or S3I201. It was 90 micromolar, and we needed to drop that number down. So we did our first change. So at U of T, the tenure process is pretty hard. So you need to have lots of projects working in order to get tenure. So we made our first change, and we replaced this oxygen with a nitrogen. And we lost all activity entirely. So the number, the IC50, shot up to over 300. So at this stage, I was thinking of excuses to get my Canadian girlfriend to come back to Scotland with me, because I thought I was getting fired. But if you recall, the whole strategy was to come off this nitrogen and build into the upper pocket of the protein. So we put the benzyl group on here. So the IC50 gained traction. It came back to 300. Paracyano 260, but tert-butyl 190, biphenyl 115, cyclohexylbenzyl 4. So we ended up with a 20-fold more potent compound than we originally saw. So this is 20 times less drug, kills the same number of cancer cells. So this is called SF1066. This is named after the student that did the reaction. So this is Stephen Fletcher, Lab Book 1, Reaction 66. So he's now a tenured prof at the University of Maryland in Baltimore. So this wasn't potent enough. Remember, we needed to get it down to 0 point something micromolar. So we did more chemistry, and we identified this compound, BP1102. And it had an IC50 of 0.8. So we were now in the low concentrations to elicit the same amount of cell killing. This is Brent Page. So Brent's a professor now. He's from Hamilton. He's now a professor in Sweden at the Karolinska Institute, which is a very famous research center in Sweden. And this molecule had no effect on cells that had no STAT3, and it had a profound effect on cells that had STAT3. It also completely knocked down STAT3 in those cells when we looked at it through cell biology. And the next stage was then to do pharmacokinetic analysis. So what is that? That's when you inject your drug into mouse models typically. And what you're trying to see is whether you can recapitulate what you saw in cells in a living system. So the first thing that you have to know when you put it into a living system is, do you actually have a therapeutic dose in your animal system? So the problem with living systems is that they have livers, and livers are the bane of medicinal chemists' life. So the liver is an organ that is chock full of enzymes that break up and digest proteins and small molecules. And they do this in the most efficient manner. They break up everything. 99% of all small molecules fail because of the liver. So Brent injected his compounds, and then he looked to see how much was left. So in this graph, you've got the concentration of your drug, and you've got time. And here's what happened to the drug. It completely disappeared. And let me just state this. So we'd spent about $1 million at this point perfecting this structure. In the analogy of the jigsaw puzzle, it fitted perfectly. It was designed and tailored for STAT3. This is what the liver did to it. It completely changed its structure. It added groups on here, it added a group here, it added a huge group down here. So when you look at the actual change in shape of the molecule, you'll recognize that that's not going to fit the same way that that original molecule was designed to fit. And in fact, it didn't fit at all. Moreover, it was completely cleared from the blood, the plasma, within 10 minutes. So there was none left after 10 minutes. So the liver metabolized it so fast that it was completely gone. So what's the solution to this? Chemistry. So chemistry can be used to look at those sites where the molecule is labile to metabolism And it can be used to introduce functionality that can stabilize the drug so that the enzymes don't recognize it and don't metabolize it. The upshot was that this molecule was created, AM1124, and this molecule had way better stability. So we had therapeutic doses of this drug in animals for 4 hours. So that wasn't good enough for us, so we actually did a wee bit more chemistry. I'm not allowed to show you the structures. We'll call it compound X. And we had some beautiful data with this compound. So what you see here is a multiple myeloma trial. So this is a bone, blood cancer. There's no cure for multiple myeloma right now. And this is an actual patient sample that was taken from Princess Margaret of recurrent multiple myeloma. So there is no cure for this particular cell line. So it's resistant to radiation, it's resistant to drugs. And what you see in this blue blob is not just the tumor, but it's actually a fluorescent marker that tells you that STAT3 is firing in this tumor. So it's a readout of activated STAT3. So this is a really cool image, and I'm just going to write that here because your idea of cool and mine's probably really different. So what you see with time here is the firing of STAT3 inside this tumor. So what happens when we treat with compound X? You can see a temporal shutdown of phospho-STAT3 inside the tumor. So this molecule is able to, in an 8-hour period, completely silence this oncogene, this protein that's driving the tumor disease. We also tested it in a recurrent medulloblastoma tumor. So this is the most common children's brain cancer. So the standard of care right now is horrific. So it's huge amounts of radiation, and it's a combination of 3 super cytotoxic drugs. One of those drugs is analogous to nerve gas that's used in the First World War. So this is the level of toxicity that these children are exposed to. And it's considered a good standard of care in that 65% of these children are cured of their disease. But the side effects of this treatment are horrific. So these children have hearing loss, they lose their teeth, they have mental impairment, and they are subject to potential recurrence of different cancers at a later stage in life. So it's a treatment, but it's not a good treatment. The other 35% are palliative, so they go into palliative care. And so what we did with McMaster with Dr. Sheila Singh was look at the recurrent model. So these are tumours that have been taken from patients that have had radiation and chemotherapy and have failed. So these are mouse brains that have been taken from mice that were grafted with the brain tumour, and we treated with our compound. And what you can see, so these are mice that have had surgery, they get brain cancer, the tumour's allowed to grow, and then we treat these mice. And what you can see is that this drug has a profound effect on the size of these tumours in these mice, and it has a profound effect on the extension of life in these mice, and it also has a profound effect on the quality of life of these mice. So I'm just going to show you a wee video. This is the mouse that's been treated with compound X, and this is the mouse that has not. Both of them have brain tumours, and you can see that the one that's been treated with our drug is happy, it's behaving like all mice do, or it's running around. The other mouse is moribund because it hasn't been treated. So it shows you that even though they both get brain tumours, our drug gives them a much better quality of life. We also went into acute myeloid leukemia. This was the start size of the tumour. At the end, day 13, the tumour grows. When you treat with our drug, it shrinks significantly. So this took 8 years to achieve to get this drug. It culminated in a deal that we did with Medici, a UK-based venture capital fund who are extremely famous now. After last week, they did, I don't know if you saw this deal, Celgene bought, so we were one of 20 projects that they brought on board, and they sold their first asset last week to Celgene for $7 billion. So our deal looks pretty bad right now. But they're a VC that are really good, and they take drugs to clinical trial, and their original fund was the Index Ventures Fund that came up with Skype and all the like, and they split off into a life sciences fund and a fintech fund, and so we benefited from this. They licensed our drug in a $22 million deal where they want to drive this drug into phase 1 clinical trials. The disease that we're looking at is AML. They are also now considering DMD for this class of compounds. We are using Jesse's Journey funding at this point to try and validate these molecules in DMD. VCs go after orphan diseases. This is their business model. And I can tell you a story just at the end, but as a result of this deal, 14 PhD level scientists are now hired in Canada. We also have developed another technology which we call DT1. It's a cancer-killing weapon which is cloaked. It's a bit like a Klingon bird of prey is my analogy. So I told my students that I was going to use this analogy. I was giving a talk in London, England to U of T alumni. They said, nobody's going to get this. So at the end of the presentation, someone came up and said, you know, the Klingons didn't develop cloaking technology, the Romulans did. And I said, I really don't care. So the idea of this technology is that you can introduce a potent warhead into a drug. Warheads that were previously not considered. And once it gets to the site of action, it opens up, decloaks, and reacts with a protein. So the upshot of this has been that we've used this molecule now to effectively cure AML. One of the issues with AML is that the cancer cells reside in the bone marrow. Most drugs don't get into the bone marrow, and so that's why AML is really hard to cure. It's why CML is hard to cure as well. And we completely suppress the tumor cells in both bone marrow and in blood. So we saw this amazing activity. It only took 6 months to do this in our current lab setup. From this, we've spun out another company, Dalriada Therapeutics. So we've already had loads of potential investment in this. So Dalriada is the ancient kingdom of Scotland and Ireland. So nobody knows what it means, and nobody can pronounce it. So it's always a good talking point. So we were in this competition, this pitch competition with Royal Bank of Canada, and the lady went up to the mic to announce the winner of the competition, and she looked at the name, and then she turned to the side, and I said to my business partner, it's us. And it was. So it was funny. My next door neighbour he's a Scotsman as well, and his name's Brian McAlpine. So the McAlpines were the kings of Dalriada, so he was really happy that I chose this name. So I'm going to say where I dreamt of being in 5 years. So we set up this system in my lab where we do chemistry, biophysics, and cell biology all in the one place. And U of T have really supported that vision, where we have a 4,000 square foot facility where the entire process of drug discovery can happen. The one thing that was missing was animal trials, and what we decided to do, I pitched this idea to the Orlando Corporation, to Carlo Fidani, where it would be nice if we had a building that made molecules. The molecules move downstairs, they can be tested biophysically against the proteins, and if they don't work, they go back up to chemistry, they're optimized until they do, and eventually they come down and they can be tested against cancer cells or any type of disease on a much larger scale. And the idea was very simple, that what we'd love is to have the preclinical trial animal facility built in so that we could be a one-stop shop for identifying molecules against any disease right up to an advanced preclinical stage. Effectively to the point where we're looking at getting private investment, $2 to $4 million to take it to an IND level to get it to phase 1. And so I'm happy to say that that's where I dreamt. So we're now building at UTM a $150 million building that is going to be geared entirely towards drug discovery. We're hiring top-tier faculty from around the world. We're looking at a cohort of around 200 PhD-level scientists working on drug discovery and producing advanced preclinical candidates. And the idea here is, one, if we're doing medchem in academia, if we're not creating drugs that people want to invest in to take to clinical trial, then we're not doing our job properly. And two, there's a real dearth of pharmaceutical industry in Canada. In fact, R&D in Canada is pretty much gone. And so we see this as a way of trying to spin out small startup biotech companies that have good, solid data behind them to garner investment and to take drugs to trial. And this is something, particularly in the orphan disease field, that needs to happen. And we're hoping that this facility is going to foster that type of mentality within Canada, the entrepreneurial mentality that sometimes is lacking in the chemistry departments in this country. So all of this led to an accidental introduction to DMD. So Andrew from Max's Big Ride came to me after we'd done this deal and basically said, do you guys look at DMD? And I was very honest and I said, look, I'd love to work in this field, but I don't think STAT3 is active in DMD, so I would have to start an entirely new program, looking at what the potential targets are and starting from square one, and I didn't have the funding to do that. But I said to him, look, what I'll do is I'll have one of my PhD students investigate the literature and see whether there's a link or whether there's some application of STAT inhibitors in Duchenne. And so it turns out that actually there's tons of links between STAT3 and DMD. And in fact, Michael Rudnicki, who Jesse's Journey supports to a large extent, it was 2 Nature medicine journals, like the top tier journals, showing that STAT signaling was involved in stimulating adult satellite cell function in Duchenne. And so it was just crazy. Actually, it's three Nature papers in the last 3 years showing that this was the case. So we couldn't believe it. And we came back and we almost felt guilty saying, actually, there's really good reason to test a STAT inhibitor. I didn't want to look like I was just trying to get money, but I was like, man, there's a great reason to try these STAT inhibitors. And so it's huge in DMD. And so Andrew is actually, we now have a full scholarship for Yasser, who I think is involved in the videos as well. He's this unbelievable young man who's just a dynamo, who's going to be working exclusively now on DMD. So we're in the process of scaling up all our STAT drugs for trial now in DMD. So we're going to look at cell culture first. We're going to look at then moving on into the animal models of DMD as well, which are in Halifax. So just to let you know, the investigations are going to be starting very shortly with our compounds. So to summarize, I hope I've shown you that you can, through very rational means, start from a rubbish compound and end up with a $22 million funded asset. And our goal is to repurpose an oncology asset towards DMD. One of the crazy things that I should also mention is that one of the other major projects in our lab is HDAC, histone deacetylase inhibitors. Guess what's active in DMD? Histone deacetylase. So we have the only HDAC11 selective drug-like inhibitor in the literature. So with the funding from Jesse's Journey, we're going to put this into this as well. So it's better to de-risk these types of projects, and let's look at lots of different molecules to see which one sticks. And just to acknowledge, this is my research group at Toronto, so I've got 35 PhDs and postdocs. It's a lot to handle, but they're actually really motivated, intelligent guys and girls that are just super driven to get drugs to clinic. And one of the things that maybe is slightly different about us in terms of academia is that we view the advancement to clinical trials as being just as important as the basic science. And all of this stems from basic science, but ultimately the basic science has to lead to a translational technology which can go into humans. And so hopefully we'll be in humans pretty shortly with our first drug. And that can be all the toxicology studies that are going to be done and paid for by the VC will de-risk and facilitate a clinical trial in a DMD STAT3 drug as well. I'd like to thank all my research sponsorship from all these and many private donors, and I can now add Jesse's journey to that list as well. And thank you for your attention, and I'd be happy to answer any questions if you have any.